Transcription and translation. How our genes code for our traits.
So did you ever wonder why you're as ugly as your dad? Well, the basic reason for that is because he gave you some genes that gave you some traits. That is, some DNA that caused you to have certain physical characteristics. But this is oversimplifying things, because to get from genes to traits requires several intermediate steps. Genes are actually segments of DNA molecules, and traits are caused most directly by protein molecules. So when we say our genes cause our traits, what we really mean is that our genes are made up of DNA, and our DNA causes us to make particular proteins, and then the proteins cause our traits. And even the DNA and the proteins can't do it alone. They need a go-between molecule called RNA to help them. The transfer of information from DNA to RNA to proteins is so well studied and so important to a geneticist's understanding of biology, it has been come to be called the central dogma. And the objective of this video is to examine how information flows in the central dogma, from DNA to RNA to proteins. Here is an illustration of how the central dogma occurs in a cell. Around the outside of the cell, you can see the cell membrane. This large, round, central organelle with DNA inside is the nucleus. Remember, the NOVA video compared the nucleus to a little castle. Inside the nucleus is DNA, which the NOVA video compared to a cookbook kept inside of the castle. That makes the nuclear envelope kind of like the mean librarian, who does not let the original book of DNA come out, though it will let occasional copies of the pages escape. How are the copies made? By this large blue enzyme, which the NOVA video compares to a scribe copying the DNA. The copies themselves are called RNA, and the nucleus does let them out of the little windows of the nucleus. For example, here are some RNA molecules that are leaving the nuclear pores. Once the RNA molecule has been let out of the nucleus, it and its compatriot RNA messages are free to float about the cytoplasm, like the recipes floating from the tower top. In the cytoplasm, the messages are read by yet another molecule called a ribosome. Like tiny chefs, these ribosomes read the recipe delivered by the DNA and use it to cook something. They make a protein. This artist showed that the strands of proteins are actually long chains of individual amino acids that have been attached together, and that's what the ribosome is doing, reading the RNA message in order to attach the appropriate amino acids. So this is an overview of how information travels from the DNA to the RNA to the protein. The title of this talk included the words transcription and translation. Those two words are involved in this process. As you can see over here on the left, in order to get from DNA to this RNA copy, a process called transcription takes place. And in order to get from the RNA molecule to the protein molecule at the end, a process named translation takes place. These two names are really apt because transcription has this word in the middle of it, script. Script means to copy or scribe, whereas translate, you may already know, means to change languages. And that's exactly what the cell is doing. It's taking a DNA template, so that means inside your genes, a section of your DNA, and it's making a copy of that DNA in the form of RNA. RNA and DNA are very related molecules. Then the cell is reading this messenger RNA molecule and using it to construct a protein. Now, proteins are made not of these nucleotide letters like DNA and RNA, it's made of a different alphabet of amino acids. And so to change from this language to this language, scientists call that translation.